Hello, everyone. Thank you for joining this installment of Boyce Thompson Institute's Breaking Ground discussion series. My name is AJ Bushy, and I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome BTI adjunct faculty member Michelle Heck, who's a molecular biologist at the U.S. Department of Agriculture, and who will be talking with us today about her efforts to combat citrus screening disease. Hi, Michelle.
Hello, good afternoon. How are you?
Doing well. Thanks again for joining us.
Thank you so much for having me.
To start us off, Michelle, could you tell us a little bit about yourself and how you got into citrus greening disease and at the USDA?
Yeah, sure. I will share my slides that I've prepared to help guide our conversation. Can everyone see my screen okay?
I can.
Great. So about me, I grew up on Long Island, New York, in a suburb right outside of New York City. And I grew up as a musician. I started playing various different instruments when I was 5 years old, and music was my life. But then when I was in ninth grade, I was invited to go on a field trip with the seniors, the 12th graders at our high school, to this place called the Dolan DNA Learning Center, which is pictured here at Cold Spring Harbor, which is about 45 minutes east on Long Island, where I grew up. And it was there during that field trip that I ran my first agarose gel. And it's a type of experiment that lets scientists separate DNA and actually look at DNA. And we were able to visualize DNA from a virus that infects bacteria in that class. And we got a printout of the picture of the gel, and I hung that picture of the gel up in my room. And I, like, everything that I thought I was going to do with my life, up until then, just was totally upended. And I was like, wow, I just want to run gels for my entire life. This is so cool. So it kind of created a little wrinkle in my plan to be a musician. But I did end up going on to college at Boston University as a music major. And I chose Boston University specifically because they had a very strong conservatory for music education, and they also had an outstanding science program. So I knew, kind of like in the back of my mind, as I explored music, if it didn't, it wasn't for me, then I could pivot to science. And I did. I graduated with my degree in biology from BU. And then from there, I went to Cold Spring Harbor Laboratory. The Dolan DNA Learning Center is the Outreach and Education Center of Cold Spring Harbor Laboratory. And so I went back to Cold Spring Harbor. They had a brand new PhD program. I was in their first class of entering PhD students. And that was really exciting to be a part of that. And it was there that I really ignited my passion for plant science and plant biology research. And so, yeah, I was a first generation student. So there was no sort of path in my family on how to become a scientist. I had to sort of figure that out. And I had a lot of great mentors along the way. And once I finished my PhD, I moved up to Ithaca, where I started my family. And I did a postdoc, on the campus of Cornell here for the U.S. Department of Agriculture, working for a USDA scientist, Stuart Gray, who is a virologist. And I basically have never left. So from there, I started applying for jobs and looked elsewhere, but no one really had this unique environment that I could find at Cornell and with the USDA. So I applied for an open position in the USDA back around 2012, 2013. And when, sort of news at these institutions about my hire and my expertise became known to Cornell and BTI, all the institutions collaborated to create a position for me. So even though I'm a USDA scientist, when I opened my lab, it was housed inside the Boyce Thompson Institute. And there are many other USDA scientists now and through the years that have been part of that BTI USDA partnership. And I have an adjunct appointment in the section of plant pathology and plant microbiology at Cornell. So I also am married with 3 beautiful kids, 16-and-a-half-year-old daughter who is very interested in science, and 2 twin boys who are 14 and interested in science and a variety of other things. So
that's great. And BU just beat Cornell in men's hockey in the playoffs. I don't know if you saw that.
Oh, that's good.
Yeah,
I don't know where my loyalty lies.
I was going to ask who were you rooting for, but
Yeah.
Cornell had a great season. BU's moving on, but Cornell's really had a great season this year. So that was
You
nice
can't
to
win
see.
everything, you know.
No,
Part of it
no.
is sort of how you play the game and your mindset, which
Yep.
applies to science research in a lot of ways.
That's a good observation. So let's talk about citrus and citrus greening disease. How did you could you tell us more about that disease and how you got into it?
Yeah. So yeah, when I was hired through the USDA, I was actually hired to work on aphid transmitted viruses in potato. And amazingly, there are a lot of biological similarities, the way aphids would spread a plant virus and the way the insect vector of citrus greening is spread. And so we're going to talk a little bit about that. And so in the early years of my hire in the agency, the agency created this program called the Grand Challenges, which are like the super massive problems in agriculture, in US agriculture. And scientists who participate in Grand Challenge research are given authority by the government to work outside their assigned research area if they are working on this Grand Challenge. And so the Office of National Programs at USDA saw those similarities and allowed me to start working on citrus greening disease, which was great. And so, you know, citrus greening disease, what you see here pictured is a very family-friendly and wholesome image. So if you think of Florida, you think of, you know, Mickey Mouse and oranges, except that the situation in Florida for the growers now is not so wholesome and positive because of citrus greening disease. The disease is caused by a bacterium, and all citrus can become infected. All types of citrus can become infected. And ultimately, several years after the infection, the trees die. And, you know, there's basically, you know, every citrus growing part of the state of Florida has the disease, and it's extremely difficult to manage because there is no cure and there is no sort of natural resistance that you know breeders can use to create sort of normally resistant plants. And so this is a familiar scene. If you drive up and down the I-95 corridor in Florida, you see these very sad abandoned citrus grove graveyards with just rows and rows of dead and dying trees. And the economic impact, you know, when you sort of put pencil to paper and you look at the numbers, it's just devastating. So the insect vector that spreads the bacteria was first discovered in Florida in 1998. But then the disease was discovered in 2005. And so what this graph shows is after the disease was detected, sort of, you know, from 2004, the percent decline in citrus production for oranges. And you could see it's over an 80% decline, the industry now. And I've read forecasts between like 14 and 16 million boxes this year, which is just a very small fraction of what you know the state normally produces. So
Wow.
it's an emergency right now. This is a state of emergency for the growers in Florida, and the other citrus growing states are at very high risk. So the disease has already been detected in California, in Texas, and other areas where citrus is grown. And so this is not something that we can sit complacently by and you know, just watch it destroy the industry in this country. But you know, for Florida, they're in a crisis situation, and when you're in a crisis, you have to think and approach problems very differently. And I had a really cool discussion with the research leader in USDA down at Fort Pierce, Florida, which is like the epicenter of the disease. Her name is Dr. Erin Roscoff, and she said, you know, Michelle, look, there are two types of scientific research. There's watch it happen and make it happen. Okay, most of us as scientists are trained in watch it happen research. And watch it happen research means if you set up an experiment, you test the hypothesis, and whatever the results are, you watch that unfold, and it's reportable. You write a paper, you learn something new, and it's important because you're contributing knowledge. But if you're doing make it happen research, and you're a scientist tasked with delivering a solution to citrus greening disease, You have to make that happen. And that requires a totally different mindset and approach to science, focused on what can growers actually use in the grove, what's commercializable, and so all of these things drive our approach to the science and interactions with our stakeholders.
That's a great way to think about it, of just starting with, this needs to be a practical solution. So even if you see something that's kind of neat, if it isn't practical, you just kind of push it to the side and save it for later, or?
Yes, you have to push it to the side, put it on the shelf. It's not that it isn't interesting or important academically, but if it isn't going to solve the problem or be able to be utilized in the way that the growers grow citrus in Florida, it can't get as much time and resources, because we don't want to take away time and resources from the things that could help. And yeah, so I'll just leave it at that.
Yeah. So what are some of the solutions being used today, or attempted solutions? What has been attempted, I guess, so far to combat this disease?
Very good question. So there is an emergency use exemption for the use of antibiotics, oxytetracycline specifically, and there is a company that sells a sprayable formulation of oxytetracycline. And there is also now approval to use the same drug, oxytetracycline, directly injected into the tree. So you essentially drill a hole into the tree and you take a syringe filled with the antibiotic fluid and you pump that into the tree with the expectation that the antibiotics are going to eventually get to the blood supply of the tree where those bacteria are found, and then treat like you would any sort of bacterial infection in humans. And there's no doubt that actually this approach of using antibiotics is very important to put these currently infected trees on life support for now. But we don't think that this is a long-term sustainable and safe solution for the industry, moving forward, and how we want to grow citrus in a sustainable manner moving into the future. And so for that, our lab is working on developing biological sources of antimicrobials that are normally found in nature. So plants and insects, they produce molecules that naturally help us fight off infection. And one class of these molecules are called NCR peptides. These are plant-derived peptides that are naturally produced by legumes. And so interestingly, when legumes grow in their root system, they interact with a bacteria, a beneficial bacteria called Rhizobia meliloti, that helps the plant fix nitrogen. And encoded in the genome of Medicago, this legume, are these small peptides called NCR peptides that the plants normally use to control that interaction with the bacterium and make sure it's staying on the friendly side, right?
Yeah.
And others in science have actually taken those peptides and showed if you apply them to pathogenic bacteria in animals, Salmonella or Listeria, for example, you can actually kill bacteria with these peptides. You see these are what's called electron micrographs. They're pictures at very high magnification of bacteria that are treated. And, the cells are just, you see this like membrane perturbation and totally cavitated, and they essentially die because they're working like antibiotics, except they are plant derived. And several members of my laboratory over the years have thought, this would be a very cool project to develop. We're providing the industry with a source of molecules of peptides that could control citrus screening. So this is Stacy de Blasio. She's a staff scientist at the USDA in my location. Dr. Laura Fleites. She now works for a biotech company down in Florida on citrus screening solutions, and we're still collaborating. John Ramsey, who is a postdoc, Stephen Higgins, who was a postdoc who moved on to a job at Battelle, and David Igwe, another postdoc who's still working in the lab on this project. So this is one area where, we hope to be able to provide growers with peptides that are plant-derived that can treat the bacteria. This way, it'll help ensure the safety of the food supply and citrus supply. But the big question is, how do we deliver these peptides,
Yeah,
right?
I was just about to ask you, is this going to be a spray thing? Because that doesn't, I just back up for a second. That doesn't seem like the greatest idea to just be spraying antibiotics all over the place. I mean, don't you risk getting antibiotic resistant bacteria, which is a huge medical problem?
100% correct. So the companies that actually supply the antibiotics, they are obligated to do very careful what's called resistance monitoring in the environment to see and make sure that you don't get that development of antibiotic resistant bacteria as a result of the treatment. So, there are some safeguards in place. But you bring up an excellent point, which is that, spraying the antibiotics, or in this case, peptides, it's not a very efficient way to get the molecules, the therapeutic molecules, into the part of the tree to cure the disease. So the bacteria live in the blood supply of the plant. And it's a vascular tissue. It's called phloem. And so that's like saying, you go to the hospital and you're septic and you have some sort of bacterial infection internally, and the doctor is going to treat you by rubbing, bacitracin on your skin. It's never going to really work well. And so we started thinking about ways in which we could develop a biological strategy to deliver peptides and other molecules to citrus. And that's really a major focus of our research right now.
So how does the bacterium get inside the phloem? I don't think we talked about that yet. How does it get infected?
That's a super good question. Actually, the bacteria are transmitted by psyllids, which are small sap-sucking insect vectors. And the psyllid vector was first found in Florida in 1998. Psyllids are extensively found now throughout California as well. And the insect has these piercing, sucking mouth parts, like a needle. And when it feeds, it doesn't chew the leaf. It inserts this needle directly into the plant vascular tissue like a straw, and it sucks on the phloem sap. And when it's taking up the phloem sap, it's also spitting out the bacteria, causing a new infection. And so the bacteria colonize the whole plant vascular tissue eventually over time. And that's really what leads to the disease and causes the plants to die ultimately, is that systemic infection. So delivery of the therapies right to that blood supply of the plant is super critical.
Yeah. So what other delivery mechanisms are you looking at?
Yeah, very good question. And that sort of brings me to the next point here. So we are developing a new paradigm for delivering plant-based therapies. And when I say we, I would like to emphasize that I am just one member of a big team of scientists now working on this. And so it wouldn't be possible without the contributions of all the team members in Florida, Fort Pierce, Florida, and other areas of the country. Several years ago, we actually had a failed project that wasn't working out well. And we thought, let's go back to the drawing board and think about ways in which we could express peptides in plants. And we came back to the best known genetic engineer in nature, which is Agrobacterium tumefaciens. And I know that sounds like a mouthful, but we could just call it agro, okay? Agro is a soil-dwelling bacterium that causes, in nature, it causes this disease called crown gall disease. And it looks like a little tumor or a bump on the side of a tree or a plant. Mm-hmm. And if you ever went hiking in the woods and you saw these like big, tumor-looking things, those are usually caused from Agrobacterium infections. So back in the late 70s, a scientist, Mary Del Chilton at Washington University in St. Louis, she discovered actually how Agrobacterium causes this tumor. It's fascinating. The bacteria have this T plasmid, and on this T plasmid is this little piece of DNA called the tDNA. And during infection, the agro inserts that tDNA into the plant genome. Okay, it takes a piece of its own bacteria and puts it in the plant genome. And in the 80s, scientists were like amazed. They were like, wow, we could take advantage of this for genetic engineering. And what they did was they removed all the genes from this tDNA that caused this crown gall to develop and put in any gene of interest. It could be a gene that, controls the height of a plant or disease resistance. And that's how the birth of transgenic plants happened, really, was the use of this agrobacterium. And so we thought, could we take advantage of this by putting back in those genes that cause this rapid cell division together with our therapeutic gene of interest, or, however many genes we want, to create like a biofactory on the side of the plant. We call it a symbiont, to refer to the beneficial nature of the cluster of cells with the tree. So essentially, this is our strategy. And before I move on, do you have any questions about that part of it?
No, I was just gonna say it almost sounds like an insulin pump that makes its own insulin. So you never have to.
Exactly. It's exactly like that, AJ. That is so, yes, it is like an insulin pump that, you could wear that delivers the therapy continuously or even on an as needed basis to a diabetic, right? And so we take our, agrobacterium with our, what we call our symbiont plasmid, which has the genes to cause the plant cells here to divide, and we create the structure. This is a picture of a symbiont on a citrus tree in Florida. And the plant cells are modified in such a way that they can grow into the structure and produce therapeutic molecules. And so this cartoon depiction shows that the symbiont cells produce and deliver the therapy directly into the tree. And so it harmonizes with the biology of the tree. And actually, the tree's vascular tissue, as this symbiont is growing, the tree's vascular tissue grow into the symbiont as it's growing. So there's an interchange between what the tree is producing and what the symbiont is producing to allow that therapeutic delivery.
That's so cool. And so you've made the symbionts, and where's the research now? Are you looking at different peptides or?
Yes, the team of scientists at Fort Pierce, led by Doctors Marco Patino and Laura Fleites, who was the postdoc in my lab, they are making all different types of iterations of symbiont constructs that express different antimicrobial peptides to see which of these are going to be effective against citrus greening disease. And they have a pipeline of greenhouse with, up to 8,000 citrus trees that are infected to evaluate these various different combinations of symbionts. And we have a few that look like they're working in the greenhouse. We have some exciting preliminary data that show, and it is preliminary, that show the symbiont can reduce the titer of the bacteria and, help to restore healthy looking plant material in those trees that develop. But what we really need to know is how even if we get it working perfectly in the greenhouse, the big question is, will it work in the field? Will it work in a real tree that is rooted in the field and that is managed commercially? And what are the limitations for that? And so right now, and just this morning, in fact, our team is developing permits to be able to conduct field trials in Florida to evaluate this. And we have a field trial site identified of slightly over 1,000 trees. And there's another government agency involved in helping us, do this field trial safely. It's called APHIS, and they have a set of rules that your site has to have all of these conditions and make sure that we can get the site prepared for inoculation of 1,000 trees. And so that's imminent. We expect to be able to do this work, coming up very shortly.
And would you need to put these on every single tree, or would it be kind of like herd immunity type of thing, where if you get so many trees, then your whole field is good?
That's such a good question. Okay. And I would say that the answer to that question depends on whether you're in Florida or in California. Okay. So if you're a grower in Florida right now, you would want to have one of these on every single tree, because your trees are likely infected. Even a new planting that's been in the ground for a year, if there's been any psyllid pressure or psyllid activity in your grove, it's likely that your tree is infected. And so the symbiont technology could be used as a cure, okay? It could be used in a way to deliver a peptide that will control the infection and alleviate the symptoms in an already infected plant. It can also be used, hypothetically, we still haven't, we're just starting testing this idea, is prophylactically. So if you put a symbiont on a healthy tree expressing antimicrobial peptides, can you prevent infection in the first place? And in that case, in California, where there is no infection yet in commercial growing areas, you may be able to only plant border rows with the symbiont. So as the psyllids are moving in, then you're slowing down the infection or blocking the infection where those trees are, and you can sort of monitor those border trees very carefully for any infections. So maybe a combination of both.
Yeah, it'd be interesting. Is there any, I'm guessing I know the answer to this question, but is there any chance that the symbionts could kind of like cure the psyllids? Like if they're sucking up, if they suck up the peptides, would that kill the bacteria in them and kind of like disinfect the insects?
Oh, that is an awesome question. So the work that we did on these NCR peptides, those plant-based peptides, oh, this was my last slide. So before, I'll make sure we get to show that. But this work here, we actually show that we screened over almost 200 of these peptides for ones that do just that. It clears the bacteria in the insect or prevents the bacteria from even entering the insect in the first place. So once we couple these with the symbiont system, and that work is in progress, we'll be able to look at that. But absolutely, peptides that are antimicrobial in the plant would be expected to be able to also have an impact on the bacteria in the psyllid. It's just a little complicated because of the biology of the insect and the way that the insect has to acquire the bacteria in the juvenile stage, the nymph, to then become competent to transmit as the adult. So the key thing there will be to make sure the application timing is when, you're just having the establishment of those insect populations early in the season.
So if they show up when they're adults, it probably wouldn't work.
Maybe not. It would be too late, because the adult tissues are already infected. We'd have to look at that carefully. Possibly. I never want to say never, but one interesting thing about these insects is that in their bodies, they have a relationship with beneficial bacteria. And these antimicrobial peptides could work against those bacteria, which have nothing to do with citrus greening disease, but they're essential for the insect to survive. And so if the peptides target those beneficial bacteria in the insects may die and may not feed properly and so on. So there could be other effects of these antimicrobial peptides that would be beneficial for management of citrus greening.
Would there be any concern of these killing any other like beneficial bacteria for the trees? I don't know. Do the trees have beneficial bacteria?
Oh, sure. The bacteria, you may have heard of the microbiome and, the gut microbiome in humans and how important the microbiome is. And people are buying probiotics left and right for various reasons. And trees have a microbiome as well, microbiota associated with their leaf surfaces, with their root, and those relationships are essential to tree health. And we think that, and this is going to be a part of the field evaluation, right? And we think that because the peptides will be, delivered in very small amounts and specifically targeted to the vascular tissue, that it may not have an impact on external interactions that the plant has with the rhizosphere, the community of bacteria in the roots. So, but those are all important things. I think the first question is, will symbionts give us a response in the tree? We want the effect to be so big that we don't need statistics to analyze it. Like we see the tree is restored and the fruit, we're getting an increase in yield, an increase in the fruit production, decrease in fruit drop, and then if we see that, then all of these other types of experiments become very important in terms of understanding how it's working, impact on the root structure, impact on insect transmission, and so on.
That makes sense. Okay, we're starting to get some questions in the chat box, but I just want to ask one more. If you look in your crystal ball, how far away do you think we might be from a cure for citrus greening?
Yeah, I don't know. Ask me again at the end of June.
Okay.
And the reason why I say that is because our preliminary data looked super encouraging, but before we can really draw conclusions about that, we need to repeat the experiment, and those experiments are in progress. And for us to evaluate the activity of a peptide in citrus, it takes about 4 months with symbiont delivery. And so we started these experiments in February, early February. And so by midsummer, we'll know if we are there or if we still have to sort of go back to the drawing board.
Did you have one last slide you wanted to show?
I did. Thank
if we
you.
saw it or
Yeah,
not.
thanks, AJ. I just wanted to mention, first of all, acknowledging the team. So this is not a picture of the whole team in Florida. I actually don't have a picture of the whole team in Florida, which I'm going to be getting in April when I go back down there. But just some members of the team here that we were doing our inoculations last month. And then my team here in Ithaca, a fantastic group of people who really drive all the innovation and the research. And it's this tight cooperation that we have between our labs and also with other labs around the country that are working, 24/7 to bring something to the growers. And these are a couple of the growers that we're working with. This is Cody Estes down in Vero Beach, Florida. And then Richard Bennett was the chairman of the California Citrus Research Board back around 2016. And Richard actually just retired and sold his farms, but because of his experience, he continues to serve as an advisory board member to our project, and having his input and Cody's input on, are what we saying and doing in the lab feasible in the way that you grow citrus? That reality check is the most important one for us. And so we're very grateful for being able to work with them in collaboration on these projects.
Yeah, well, thanks to you and thanks to everyone you're working with for trying to save our citrus. It's important.
Thank you so much, AJ.
Okay, we got some questions in the chat box. If anybody has questions, now's your time to throw them in there. First one is from Ved Prakash. Is there efforts to control the psyllids directly, so the insects?
Yes, absolutely. So we have another project in my lab that was also a brainchild of Laura, Dr. Laura Fleites, which was to use what are called psyllid neuropeptides. These are small peptide mimics that these insects normally produce, but they control everything about their physiology and behavior, from feeding to flight, reproduction, water balance, and so on. And we've partnered with a chemist in Texas to synthesize these and found ones that actually are highly lethal to the psyllid. So we're working on peptide-based solutions to target the psyllid as well. For growers in Florida, psyllid management is still extremely important. You may think, well, if every tree is infected, do we really need to control the insect vector? And the answer is yes, because the plant immune system can only sustain so much, and everything you can do to sort of help alleviate that stress on plant immunity will help the trees divert that energy to fruit production, right? So it's important still, I think, to have psyllid management as a key component of this, and we are looking at that.
Currently, so are they using regular pesticides or insecticides, I should say?
Yep, insecticides are used. Some growers use them. It would depend on their management practices, if they're an organic farm versus a non-organic farm. But they have a range of options for psyllid control. And it really would depend, the timing of those applications would depend on when the tree is flushing and when they would most likely see insects.
Okay. Next question from Kamal Tiagi. Are these peptides, do they have any effect on citrus fruit quality?
Yeah, that's a great question. We hope not. We don't know yet, but that's something we'll evaluate. If we see any of the peptides result in, like I said, a large effect on citrus tree recovery, against citrus greening, then all of these other experiments become very, very important. So, the first bar is, can we cure the tree from citrus greening? We don't necessarily have to eliminate the bacteria, right? As long as we can restore the tree back to health. For Florida anyway, if we can restore production, then there's so many other experiments then that rise in their level of importance, such as, what about the juice quality and so on? Great question.
Yeah. And one from Gary Horvath. Hi, Gary.
Hello, Gary.
Could the residue of the peptide in the fruit and then after harvest, could it affect the gut bacteria in humans?
Excellent question. Our product, we are hoping to be able to register our product through the EPA as a biopesticide. We didn't really get to talk about this much, but there are sort of two tracks in terms of using a new, input in agriculture: biopesticide and synthetic. And so, our technology checks all the boxes for biopesticide. And as a part of that registration, we will have to measure residue, okay? And there are a lot of ways in which we can control residue. And so evaluating that will be a key component, so that any residue is below, legally acceptable tolerance or zero residue, right?
Yeah, I guess there'd also be a question of how much gets into the fruit. Right.
Yeah, the symbionts, they our preliminary experiments show that, at least like in terms of what gets into a leaf, it's very hard to detect. It's very little. And so you don't really need a lot, like as much as if you were going to do a soil drench or topical application, you would have to put much, much more in there. And that's where you really have concerns about residue. But if you're delivering something directly into the vascular tissue of the plant, you don't actually need quite as much. And so we hope, alleviates the concerns or problems that we will have with residue. But that will be a key component. Residue analysis will be a key component of what the commercial partner does to advance Symbiont technology to a commercial product.
That makes sense. From Dan Geisey, I hope I pronounced your name right.
Dan Geis, yeah. Hey, Dan.
Dan Geis.
Yep.
How do you initially know if a citrus tree is diseased? Good
Boy,
question.
That is a great question. Early detection is a major challenge, okay? Because you can have one part of the tree that becomes infected. So a single infected female can come, and the females and males, they feed on this tender flush tissue, the new, very young tissue of the tree. The females lay her eggs, and those eggs develop into nymphs, and those nymphs develop into adults. If that female is infected, when she feeds on that infected flush, like 60 to 70% of her progeny are going to ingest the bacteria right from that infection point, and then have it to spread. So it could be years before the tree is like fully systemically infected, that you could like pick any leaf and say, with like doing this method called PCR, where you could detect the bacteria DNA. It could be years, okay, a couple years, 3 years. And so how do you know if the tree is diseased? Very hard. The way that the industry does it now is using this technique called PCR, where it relies on detection of the bacterial DNA. But it's not good enough, because the bacteria are not evenly distributed. And how many leaves do you sample? A citrus tree canopy could fit into this, a large conference room, right? And a small conference room. And so how many leaves do you pick before you say, yes, this tree is not infected? And so the Citrus Greening Research Board early on was very interested in like early detection methods. Could we tell by plant immunity? Could we use imaging through, satellites? And could we use dogs? There's even a program that the USDA was involved with, the same dogs that detect explosives can detect. You could train a dog to detect an infected tree. Wow. And in fact, my colleague Marco Patino, he showed that if you take a single protein from the bacteria and you express it in another plant, the dogs will recognize that plant as an infected citrus tree. Like, wow. Their noses are extremely sensitive. So big question. I can't say that there's a great answer yet for early detection, but because it takes a long time for those symptoms to show up.
Next question from F. Sessoms, I believe. Can the peptides be released by the roots and affect the root rhizosphere microbiome? So we kind of talked about that a little bit.
Yeah, good question. Again, that's a question for the future. If we see a peptide that has a dramatic effect on the tree's ability to produce fruit and get, restored from citrus greening, that is the type of question that we will go after. Based on how we know things move in plants, how peptides and molecules move in plants, we think delivery through the symbiont would mitigate leakage of those peptides through the root, but it's definitely something we're going to have to measure, specifically measure for the EPA registration process. So we realize that and we'll work towards that.
Yeah, this next question is probably kind of related from Patrick Kinder, has two. How long could the delivered peptides persist in host plants before being degraded? We'll go one at a time.
Yeah, very good question. So with the symbiont delivery method, those plant cells that are, growing in that symbiont structure, if we wanted to, they could continuously produce the peptide. So that, even if there was peptide turnover and peptide degradation, you would always have a fresh supply of the peptide being made in the symbiont. And so that's one advantage, is that you wouldn't have to have multiple applications of your therapeutic through the growing season. And in terms of, BT, the BT toxin.
Oh yeah, I hadn't asked that one.
Oh, sorry, that was I was looking at that. Go ahead.
Okay.
Yeah.
I was going to ask a quick follow-up on that. I guess, is it if the peptides aren't degraded very fast, and could there be a big buildup from the symbiont? Like how? I guess that's a question for the future, but is that a concern?
Yeah, I mean, we would be something we'd have to measure. These peptides are not really modified for increased stability. And so I don't think that they, just knowing what we know about peptide biochemistry, I can't see that they would build up. But for sure, we would, peptides that show promise in the Grove in these field trials, we're going to develop specific ways to measure them, using antibodies or mass spectrometry, and then address all of these really important questions.
Because most peptides would naturally degrade anyway, right? After a certain amount of time.
Yeah. Yep.
So the next question, the BT question, what are some advantages and disadvantages of peptides over the traditional BT or Bacillus thuringiensis based methods?
Yeah, there's a scientist down at the University of Florida. Her name is Dr. Bryony Bonning, and she's actually working to optimize BT for specifically targeting the psyllid. And so it's possible that we would be able to partner with her to deliver her, her BT peptides or BT proteins through the symbiont. And that's something, we can certainly look to do in the future.
Yeah, I tend to think of the BT toxins as they usually target like moths and things, don't they? Or
yes, chewing insects. So she has her research is focused on developing ways to target those BT toxins specifically to the insect gut, the psyllid gut, because those, the psyllids are a little bit different physiologically than, let's say, a caterpillar. So,
right.
Yeah.
Because they're sucking it up and not chewing.
Yes, exactly.
Yeah. Okay, one from Monique Rivera. In California, there was a lot of talk about soil microbiome to help the tree resist infection. What do you think about this? Can tree health overall outcompete the disease? That's a good question.
Yeah. Hello, Monique. Nice to see you. Thank you for coming. So, one of the things to sort of help give some people context, there's this phenomenon known as the survivor tree. Okay, so if you go into an orchard that's been infected for several years and you see one tree that's doing really great, why is it not succumbing to the disease? And one hypothesis is that there's something about the root microbiome or the collection of microbes in the root architecture associated with that plant that's providing some external protection. In terms of what I think about this, with microbiome research, it's very hard to test the role of individual microbes or even, subsets of the community on tree health, because there are many partners in the soil that you cannot culture. Okay, they only survive in the context of their association with the plant or with another microbe. And so direct tests on, directly testing these hypotheses are very, very challenging. And I think if there is something to that, perhaps, research with the symbiont could help to provide support for that microbiome. And so, we talked about peptides, delivering peptides through the symbiont that kill the citrus greening bacteria. But what if we could deliver peptides through the symbiont that promote association of beneficial microbes with the root? That's really my thoughts on it. Very hard to sort of test this empirically because there are so many microbial partners of plants that you cannot culture. And in terms of can tree health overall outcompete the disease, I just don't see that as, being very likely. But based on what we know about the infection, when the trees become infected because of the pathology and what happens during the progression of the disease, an infected tree is going to decline.
That's a good answer. Next one from Gonzalo Silva. Are you using smart traps to catch psyllids, and could you detect the viruliferous ones as an early warning system before employing the symbiont strategy?
Yes, the psyllids are a sentinel for this, okay? They're the canary in the coal mine. So if you find an infected psyllid feeding on your tree, there's a good chance that it came from, it's 100% chance it came from a nearby infected tree. But depending on the life stage, there's a good chance that your tree is also becoming, inoculated. I would say not infected, it's becoming inoculated with your bacteria. And so surveillance of psyllids is very important in places where, like California, where the disease has not fully emerged in commercial production areas, okay? Because if you're finding infected psyllids in your grove, you should be very worried that those psyllids have come from an infected tree source. For Florida, psyllid surveillance, like the time has passed, okay? Because so many trees are infected, basically most of them. And so if you're going to find infected psyllids everywhere you look. And I have a graduate student in my lab, Marina Mann, who is pictured here, who went down to Florida in 2018, and she mouth aspirated almost 5,000 psyllids from across 4 different groves and showed that, you can find infected psyllids in all of them.
Wow. And from Alan Renwick. Hi, Alan.
Hello, Alan.
How do the insects find the tree? Is there any way to interfere with the behavior of host selection?
Yes, there are people working on different lures to interfere with that. And we think, one day symbiont technology may be able to be used in that manner if the symbionts are engineered to express pheromones that interfere with insect behavior. And there are groups from University of Florida that showed infected citrus trees release a compound that makes the tree like a beacon in the grove. And I think that was maybe the title of his paper or something like that. And those infected trees are very attractive to insects. And also they're predators, amazingly, the predators of the insects. So, it definitely, there's, this manipulation on the part of the bacteria that helps to promote its spread in a larger ecological context. So the bacteria manipulate the tree in many ways, in the color, in the volatiles that are given off to attract the insects, which then promote further spread.
That's fascinating to me, how it's this intricate, all 3 of them, they're all, working to spread themselves.
Yeah.
Very neat. Okay, that's the last question in the chat, but I have one more that I was wondering. Could this symbiont approach be used to combat other crop diseases?
Boy, we sure hope so. I mentioned all the different permutations that of the constructs, the symbiont constructs that the team at Fort Pierce is making with different, vectors and antimicrobial peptides and movement signals and so on, right? And so once these are made, we can test them in a wide range of crops. For example, grapes that are infected with Pierce disease. Pierce disease is a major problem in California. The growers spend on average of California $100 million a year to mitigate the damage caused by Pierce disease. So that's our next target after citrus screening. And then there's a related disease called olive quick decline. Okay, and this has had a devastating impact in Italy. More than 20 million of Italy's olive trees have been ripped out of their 120 million trees, and as part of their control measure. So, partnering with groups that are interested and able to work with us to expand this technology in those areas, it's important. There's also cherry X disease, which is a big problem in the Pacific Northwest. The disease is caused by a different type of bacteria called the phytoplasma, but same thing, transmitted by an insect, lives in the plant vascular tissue. So we can evaluate all of this existing technology that we're developing for greening could be evaluated very readily in these other systems.
That's great.
Cool.
So this could have uses far beyond just citrus. That is great to hear.
Yeah, we hope so.
Yeah. So one last question, one more popped up in the chat box from Ved Prakash. Is there a difference in the infectivity by psyllid if the psyllids is also infected with Diaphorina citri associated C virus?
Yeah, good question. The short answer is I don't know the answer to that specifically for that virus, but psyllids, like other insects, can be infected by a range of different viruses. And it's exciting that you asked that. Part of the research that goes on in my lab, led by my graduate student, Stephanie Priesing, is understanding how co-infection with insect-infecting viruses alters the ability of these insects to transmit plant pathogens. And certainly in aphids and plant viruses, my lab has published on this, and Stephanie's research has conclusively shown this. But there are other groups, I think, at University of California that are looking closely at that specific question, but my group is not with psyllids anyway.
Okay, I think that's the last question. Thank you again, Dr. Michelle Hecht, for taking the time today to chat with us. And thank you everyone out there who joined us for this virtual chat. This was awesome. This was so good. Thank you so much, Michelle. And
thank you very much.
Seriously, thank you everyone out there. You asked amazing questions. This is always fun. And if you have any follow-up questions, please feel to reach out to the communications department at communications@btiscience.org. And I'm just going to put a bunch of links in the chat box right now that I'll be mentioning. You can also see replays of all 31 of our, and just on Mike's slide, of all 31 of our previous Breaking Grounds on our website at btiscience.org/breaking ground discussion series. There's some dashes in there. And you can read more about BTI's current research and many other neat stories about BTI Science in our annual report at btiscience.org/annual report. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. If you'd like to make a gift to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. And as you probably see on the slide, this is usually where I say, hey, go online, register for our next guest. But the next event will be TBA. After 4 fun years here at BTI, I'm moving on. I've taken a new job, which makes me extra happy. I finally got you on here, Michelle. I'm so glad to have gotten you to be a guest before I left. So thanks again.
The pleasure is mine, and it's been great working with you all these years. I wish you all the best in your future endeavors, and we will certainly miss your energy and enthusiasm and tenacity around Boyce Thompson Institute.
Well, thank you, Michelle. And I'll definitely be keeping an eye on what you're doing because it's really cool stuff. It's always
maybe soon when you're going to buy your citrus in the supermarket, it'll say made with Symbiont technology. And wouldn't that be cool?
That would be very cool. That would be very cool indeed. So everyone stay tuned. The communications department I'm sure they will come up with some plans for whatever the future of Breaking Ground or other video series and all kinds of other stuff will be. So stay tuned for that. And thanks again, everyone out there. Thank you, Michelle, for your interest and support of BTI, and have a wonderful day and be well.
Thanks a lot. Bye, everyone.
Bye-bye.